So, have you ever wondered what a microbiologist actually does all day? I mean, yeah, you probably picture someone in a white lab coat staring into a microscope, and you're not wrong. But there's so much more to it. They're part detective, hunting down microbial culprits. They're part warrior, figuring out the best way to defeat them. And believe it or not, they're part mathematician, calculating the size of an invisible army. Today, we're going to crack open their toolbox and see the essential skills they use to protect all of us from a world we can't even see. All right, let's dive in. Here's the game plan. First, we'll meet the cast of characters in the microbial menagerie. Then, we'll look at the weapons they use in the battle against bacteria. After that, we'll see how they become disease detectives to track an outbreak in real time. And finally, we'll get into the surprisingly cool math they use to count a microscopic army. Okay, first things first. Every good detective needs a rogues gallery, a lineup of the usual suspects. For a microbiologist, that means knowing the potential troublemakers out there. We're going to start with a group called eukaryotes, and really, all you need to know is that these are the more complex microbes, the ones whose cells have a little command center, a nucleus. I mean, the microscopic world is absolutely teeming with life. It's this huge cast of characters with all sorts of different shapes, sizes, and, well, motives. So the first job, the absolute number one priority, is to figure out who's who. How do you tell them apart? And, the real question is, which ones do we actually need to be worried about? Let's kick things off with mycology, which is the study of fungi. Now, these guys can be super useful. Think about Saccharomyces, the yeast that gives us bread and wine. But a lot of them are what we call opportunistic pathogens. It's a great term. They basically just hang around waiting for our body's defenses to get weak, and then they strike. A perfect example is Candida albicans. That's the fungus that causes thrush. Next up in our lineup, the helminths, or as you probably know them, parasitic worms. Yeah, these are complex, multicellular animals. You've got trematodes, also known as flukes, which are these nasty leaf-shaped worms that can cause some serious organ damage. And then you've got the cestodes. You almost certainly know them as tapeworms, which famously like to set up shop right inside an animal's gut. And finally, let's meet the protozoa. These single-celled organisms are absolute masters of disguise. They can switch between an active feeding stage called a trophozoite and this tough, dormant protective cyst stage. And they're responsible for a huge range of diseases, everything from giardiasis, a pretty common intestinal parasite right here in the US, all the way to malaria, which is the world's #2 pathogenic killer. And here you can see it laid out, plain and simple, a direct line from the microscopic suspect to the disease it causes. This is really the foundational knowledge a microbiologist uses every single day. They have to connect a name they see under a microscope to a real-world illness, from a common fungal infection to a devastating global threat like malaria. So, once you've identified your bacterial bad guy, what's next? How do you actually fight it? Well, that brings us to the second drawer of our toolkit, the weapons and tactics used in the ongoing war against bacteria. This right here is the Kirby-Bauer test, and it's a brilliantly simple and super effective technique. First, you spread bacteria all over a plate to create what we call a lawn. Then, you take these little paper discs, each one soaked in a different antibiotic, and you place them on that lawn. You let it incubate for a bit, and then you just look for the results. And the results are actually so easy to read. See that clear halo around a disc? That's called the zone of inhibition. It's basically a kill zone where the antibiotic seeped out and wiped out the bacteria. A big zone means the bacteria are susceptible. The drug works. But if the bacteria grow right up to the disc with no clear zone at all, that's resistance. And that means that specific antibiotic is completely useless against this infection. It's a pretty chilling preview of why antibiotic resistance is such a huge global crisis. So, and this is the crucial point, this isn't just some abstract experiment you do in a lab. The results from a Kirby-Bauer test go directly to a physician. It tells them which antibiotic is actually going to work for their patient, guiding their prescriptions, and playing an absolutely vital role in treating infections and, saving lives. Okay, so we've looked at fighting a microbe inside a single person. But what happens when a disease starts spreading through a whole community? Now we have to zoom out, from the patient to the entire population. And this is the third tool in our kit, epidemiology, the work of a disease detective. The very first thing epidemiologists track is something called morbidity. It's a really straightforward but vital piece of data. It is simply the total number of people who get sick with a particular disease. This is one of the key stats you'll see in the CDC's Morbidity and Mortality Weekly Report. And right alongside morbidity, they track its tragic counterpart, mortality. This is the count of how many people have actually died from the disease. You put morbidity and mortality together, and suddenly public health officials have a really clear picture of the impact an outbreak is having. A huge part of this detective work is the hunt for the index case, or as you've probably heard it called, patient zero. This is the very first person identified in an outbreak. Finding them is like finding the source of a river. It's absolutely crucial for understanding how the disease is being transmitted and for tracing who else might have been exposed. Now for what might be the single most important number in all of public health. It looks so simple, right? But this little expression called R0 is the thing that keeps epidemiologists up at night during an outbreak. So what on earth is it? Well, R0 is the basic reproduction number. It tells you, on average, how many other people one single sick person will infect in a population that has no immunity. If R0 is less than 1, the outbreak just kind of fizzles out. But if it's greater than 1, that means each case is generating more than one new case. The disease is spreading, and you are officially in an epidemic. And now for our final tool. See, underpinning all of this detective work, all this battling, is one fundamental skill: the ability to count things you literally cannot see. This is where the lab coat meets the calculator. I mean, it sounds impossible, doesn't it? If you put a single drop of really concentrated bacteria on a plate, you just get a solid mat of growth, totally uncountable. The secret is this really clever technique called serial dilution. You just systematically dilute the sample again and again and again until you get the concentration down to a level that will actually produce a countable number of colonies. Once you've got a plate with a number of colonies you can actually count, it all comes down to this powerful little formula. You take the number of colonies you counted, you multiply it by how much you diluted it, and then you divide by the tiny volume you put on the plate. And this gives you the original concentration in CFU per milliliter. And CFU just stands for colony-forming units. It's a fancy way of saying the number of living, viable bacteria that were in that very first drop. And the results can be absolutely mind-blowing. For example, let's say after all that dilution, you count just 112 colonies on your plate. Well, you plug that into the formula, and you can discover that your original sample contained over 1 million colony-forming units per milliliter. It's an incredible way to measure a massive invisible population from a tiny countable sample. But see, serial dilution only gives you a snapshot in time. What if you want to watch the population grow, like a time lapse? For that, you need a different tool, a spectrophotometer. It basically just shines a beam of light through your liquid sample. The more bacteria there are, the cloudier or more turbid the sample gets, and the less light makes it through to the other side. It's a really clever, non-destructive way to track growth in real time. But all that data is pretty useless if you can't communicate it clearly. To tell the story of that bacterial growth, microbiologists rely on the simple, powerful, unbreakable rules of a good graph. You need a clear title, properly labeled axes, and consistent measurements. You do that, and you can turn a boring list of numbers into a powerful visual story, showing exactly how quickly an invisible army can multiply. So there you have it. Identifying the suspect, choosing the right weapon, tracking its spread, and counting its numbers. This is the Microbiologist Toolkit, and these are the skills that stand between us and a very dangerous invisible world. They allow us to see, to measure, and to fight back. And it really makes you wonder, right? What will the next tool allow us to see? What other invisible worlds are we just now finally learning how to measure?